Good morning, students. Now let us start with the introduction of carbohydrates. Carbohydrates are a group of naturally occurring carbonyl compounds which are made up of aldehydes or ketoses, which also contain several hydroxyl groups. So apart from that, these are the major carbohydrate sources, and this is the major energy source and easily oxidizable in nature. And these carbohydrates are soluble, of course, in water and also sweet in taste. That's why you call it as generally as sugars. In the major glycolysis pathway, the glucose, which will be converted into pyruvate molecule. So one glucose molecule will be converted to pyruvate molecule. And in case if excess of carbohydrate or excess of glucose is taken in the diet, that will be converted into glycogen by the process of glycogenesis process. And also the glycogen can be degraded and it will be converted to glucose with the help of glycogenolysis process. This occurs during starvation or less carbohydrate in the diet. And also glucose can be synthesized from non-carbohydrate sources by means of gluconeogenesis process under the prolonged starvation condition. So the prolonged starvation leads to the formation of new glucose molecule from non-carbohydrate sources also. And glucose also has another pathway, which we call it as pentose phosphate pathway or HMP shunt. This enables a glucose 6-phosphate, which is a derivative of a glucose, to get converted into ribose 5-phosphate. And this ribose 5-phosphate will be used to synthesize nucleotides and nucleic acid and nucleotide-derived coenzymes too. And this reducing equivalent has very important function in so many pathways. Wherever we have to stabilize the free radical, there and all NADPH will be used, especially in stabilizing the RBC membrane and also for the detoxification mechanism in the liver, which is happening. So this will be the general fate of carbohydrate, in which the glucose, if it is excess, will be converted to glycogen, and also the glucose, which can be converted to pyruvate by means of oxidation process. And in case of aerobic oxidation, we get pyruvate. In case of anaerobic oxidation, it leads to lactate. And from pyruvate, it will be converted to acetyl-CoA. And from acetyl-CoA, you have different sources or different ways in which it can be interconverted. From acetyl-CoA, fatty acid also can be synthesized, or acetyl-CoA can enter into TCA cycle, and through electron transport chain, we get ATP. And the complete oxidation of acetyl-CoA leads to the production of water molecule and carbon dioxide. And of course, the glucose also will enter into pentose phosphate pathway, which leads to the formation of pentose and also the other sugars, which are all of biological importance. Let us now start with glycolysis process. Of course, glycolysis in the sense, glyco is sugar, lysis means the lysis of sugar molecule. And in most of the times, this will be majorly taking place in brain, kidney, medulla, and rapidly contracting skeletal muscle cells. And some of the cells like erythrocytes, sperm cell also depend on glucose as a major metabolic energy source. Of course, already we have studied aerobic condition, you get 2 pyruvate, whereas in case of anaerobic condition, 2 lactate will be produced. So further fermentation of the lactate by means of microorganism will also lead to alcohol, and thus you call it as alcoholic fermentation. So now this particular pathway is composed of the glycolysis, and in which the glucose will be converted to 2 pyruvate molecule. The entire glycolysis process has been divided into 2. The first 5 reactions of glycolysis and the next 5 reactions of glycolysis. The first 5 reaction of glycolysis, you call it as investment phase. The next 5 reaction of glycolysis, you call it as payoff phase. In first 5 reaction, ATP will be invested, and the next 5 reaction, ATP will be harvested either as substrate level phosphorylation or through the reducing equivalent, which is formed in the reaction. Let us just go through the steps which are involved in the glycolysis process. So glucose will be converted to glucose 6-phosphate by means of hexokinase, where ATP will be utilized. So this is the first priming reaction where we invest the first ATP molecule. And then the glucose 6-phosphate is converted to fructose 6-phosphate by means of phosphohexose isomerase. So isomerization reaction takes place, and again fructose 6-phosphate is converted to fructose 1,6-bisphosphate by means of phosphofructokinase 1. And here also ATP will be used for the phosphorylation, and this is the second priming step where we invest the ATP. And now the fructose 1,6-bisphosphate is cleaved into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by means of aldolase enzyme. And this makes a 3-carbon compound, so 6-carbon compound to be cleaved into 2 3-carbon containing compound. And now this glyceraldehyde 3-phosphate and dihydroxyacetone phosphate can be interconvertible by means of triose phosphate isomerase. And now the glyceraldehyde 3-phosphate will enter into the next 5 reactions of glycolysis where we get energy. Glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate by means of glyceraldehyde 3-phosphate dehydrogenase, where you produce reducing equivalent, and this can enter into ETC cycle so that ATP will be produced. And from 1,3-bisphosphoglycerate, you will be getting 3-phosphoglycerate by means of the phosphoglycerate kinase enzyme, where we produce 2 ATP molecules. And from 3-phosphoglycerate to 2-phosphoglycerate by means of mutase enzyme, and 2-phosphoglycerate to phosphoenol pyruvate by means of enolase enzyme. And phosphoenol pyruvate will be converted to pyruvate by means of pyruvate kinase enzyme, where you get 2 ATP molecules. So overall, in the first 5 reaction, we invest ATP, and next 5 reaction, we actually harvest the ATP molecule. So according to the energetics, almost 2 ATP molecules we are investing, which means the glycolysis consumes 2 ATP molecules, and you gain 4 ATP molecule, because from glyceraldehyde 3-phosphate, the reaction will become 2,2 because 3-carbon compound, 2 molecules are formed. So 4 as such as ATP will be formed by means of substrate level phosphorylation, and 2 NADH reducing equivalent which formed, which can enter into ETC cycle. So 2 into 3, around 6 molecules of ATP will be obtained from reducing equivalent. So overall, the net gain of ATP will be 10 minus 2, which is 8 in case of aerobic oxidation by means of glycolysis. So the next class we'll start with anaerobic glycolysis.